Hello, everybody, dear friends, attendees. I'm going to talk about one of our recent publications, which has as title The Energy and Metabolic Pathways in Trefoil Factor Family Member 2, or known as TFF2, knockout mice. I am Abdelaziz Ghanemi from Canada, and these are the names of our team. I would just start by a general introduction and the background of what we are doing. For the TFF2, it's a protein that protects the gastrointestinal mucosa. We have previously shown that TFF2 knockout mice are protected from high-fat diet induced obesity. And these TFF2KL mice had smaller body weight and fat mass, but more lean mass. They have a higher gross energy intake. They have a higher energy expenditure summarized within the locomotion and UCP1 in brown adipose tissue, but it's an indicator of the heat production or the energy expenditure within brown adipose tissue in form of heat. The carcass, they have also a negative carcass energy gain, and they have an increased fecal energy excretion. All that compared to the wild type mice. So the question is, how are these TFF2KL mice protected from high-fat diet-induced obesity? The method we have used is that we divided our mice into 4 groups, depending on the diet, low fat or high fat, and depending on the genotype, wild type and knockout. So we have 4 groups: low fat, wild type, low fat, KO, high fat, wild type, high fat, knockout. Each group has between 7 and 9 mice. And the diet intervention was for 12 weeks, and it starts at the age of 16 weeks, the age of the mice. After that, we sacrificed the mice and we did measurement of body composition and tissue weights, serum analysis, gene expression, as well as protein expression via Western blotting. The results, I start with this table that shows that TFF2KL mice had lower levels of blood glucose as well as serum leptin, triglyceride, and glycerol. They also report that we have an increased expression of genes related to glucose and lipids uptake and utilization in the skeletal muscle of the knockout mice. We also have an increased expression of proteins related to lipid uptake and lipid utilization also in the skeletal muscle of knockout mice. Always in the knockout mice, we have decreased expression of energy metabolism related protein in their liver, always compared to wild type. We have also decreased expression of NOR77 in the adipose tissue of knockout mice, but only within high-fat mice, high-fat fed mice, which is interesting because it shows the combination effect of both genotype and diet. As discussion, we could summarize what we have obtained in this study, and as well as the study obtained by our research group before that. Putting all together, we can see that the knockout mice compared to the wild type mice, they simply have an increased energy intake, yes, but they also have an increased energy expenditure as well as an increased energy excretion. measured, as we said previously, in triglyceride within the fecal excretion. If we look into the different metabolic tissues, we can find that within the skeletal muscle, they also use more lipids, more glucose, which is the same within the liver as well. So the total effects are taken together, both the brown adipose tissue, the liver, as well as the skeletal muscle, all together shows that these knockout mice, TFF2 knockout mice, use more energy. Take more energy, yes, but use much more energy compared to the wild type. So even with a high-fat diet, they still do not develop obesity. Conclusion is that our previous and present studies have characterized protective roles of TFF2 deficiency in the high-fat diet-induced obesity and highlighted the TFF2-related pathways as potential functional genomics-based molecular targets to develop novel therapies. Within this context, I would like to clarify an additional point is that why we initially have chosen this TFF2 gene to explore. We have previous study in which we have given 3 type of diet or just 2 type of diet, with the third one is a reference, which are fasted mice. The second condition was low-fat diet. The third condition was high-fat diet. So the mice that were high-fat diet have an increased TFF2 expression, not only compared to the fasted mice, but also compared to the low-fat fed mice, meaning that this TFF2 actually is specifically stimulated, or its expression is specifically stimulated by the high-fat diet and not the low-fat diet, which would make it of a high interest. Why? We all know that for glucose, we have, for its metabolic regulation, we take an amount of glucose, we have the development, we have an increased secretion of insulin. That's known. But for the lipids, we do not have similar signal or similar hormone. So this could be the closest thing we have so far, meaning that it would be the response to a high fat. I mean the acute response, not the chronic. The chronic one we could talk about leptin, but I mean the acute one. So it could be a signal, or maybe exploring the pathway behind it would lead to discover such signal. It would be important if that signal could stop the food intake and control the appetite, which would have as an impact to reduce the food intake. This is the reference of the published paper in which we have detailed all what I have just explained. We acknowledge our collaborators and the financing organisms. And thank you. These are my emails. Wish you the best. And in all cases, we will have the next presentation together, because I will be presenting the next topic.
Thank you, everyone.
Is that for today, Alice?
Yes, Mr. Krishna.
Okay, so we can wind up, right?
Yes.
Okay. So, as on a conclusion note, I would like to thank all the keynote speakers for all the 3 days who had been a part of our self-time webinar. A special thanks to Dr. Sinarik, UNESCO chairperson, and all the keynote speakers, Dr. Elvisa, Dr. Soha, Dr. Aslagiri, Dr. Prasanna, Dr. Arun Chapel, and all those who had been part of our all 3 days. And we would also thank all the participants who had attended these webinars. Thank you. If you have any queries, you can reach to us by all the mail address we have mentioned in the chat. You can note it down and you can if you can directly drop a line to the mail IDs mentioned in the chat. Thank you each and everyone. Have a great day. Bye bye.